Well, okay, so it's my dubious pleasure of kicking off this series. And what I'm going to share with you in the next 5 hours, don't worry, will be some of my insights in immunofluorescence and imaging artwork instead of artifacts that I have accumulated over the last 30 years, unfortunately. Okay, so the problems that we are facing are not new. So Mr. Henry Baker, whose statement you can read here, already said in 1742 that truth alone is the matter that you search after, and if you have been mistaken, let not vanity seduce you to persist in your mistake. So vanity persisting in mistakes. So what contributes to this is, of course, bias. So, you want to prove your pet theory. So there's nothing I can help you with bias. So you have to use other means of checking that. But what I will cover in the next half hour is technical pitfalls and experimental pitfalls, especially immunofluorescence microscopy regarding that you have to be careful about in order to come to a conclusion that's scientifically valid and as good as you can do it with the reagents and the instrumentation that you have at hand. So what we all want to do is we want to produce pretty pictures, but we are not the Department of Aesthetics, so these pretty pictures should also be scientifically valid. And what you can see here is one of my beauties. So it's a quadruple stained cardiomyocyte. And you can see on the right-hand side the single channels in black and white, and I hope you can appreciate that the patterns look different. So we can visualize 3 different cytoskeletal elements using this technique, and in addition, we visualize the nuclei. Now, when I say I'm doing quadruple immunofluorescence, I'm already cheating a bit, because what I'm actually doing usually is double immunofluorescence. So usually most people combine a mouse monoclonal antibody that recognizes some protein and a rabbit polyclonal antibody, or these days also monoclonal antibody that recognizes some protein. And usually people carry out indirect immunofluorescence, so they use a secondary that recognizes the mouse antibodies and that carries a particular fluorochrome, and they use a secondary that recognizes the rabbit. And usually the 2 other colors come in by staining the nuclei with stains like DAPI, and in my case, what I like to do as a counter stain is a stain filamentous actin with fluorochrome conjugated phalloidin. So, what does Auntie Elisabeth say? Well, so this is your take-home message. So, and I will spend the rest of the talk explaining what I mean about this. So let's start with point number 1. Crap in, crap out, no matter how much a microscope costs. Well, unfortunately, that's how it is. And so your reagents will contribute to the success of your experiment. Now, your reagents for immunofluorescence will often be, for immuno stuff, will be antibodies. And there are good antibodies and there are bad antibodies, and there are antibodies produced by a company that's called Holy Cross. And here you see one of these examples of one of their products. So this is a goat antiteletonin antibody, and what you can see here in the black and white and the green in the overlay, we see nuclear signal, we see some other signal here. And this is already a bit unexpected when we know something about the biology of teletonin, because teletonin was initially cloned in Italy because it was the RNA that was enriched in muscle. So it was the highest expressed RNA in muscle, so which makes it already suspicious that these non-stripy cells show a signal. And subsequently, it was shown that teletonin is a Z-disc protein, which, when we counter stain for myomycin, should give alternating stripes, which, this doesn't really do. So this is a commercial antibody. So if you were naive, you might think, well, it must work. But then what I always recommend you to do a Western blot, because you may find out things that you were not expecting about your immunofluorescence reagents. And in your immunofluorescence, you will only see where your fluorochromes land. So you have no other indication whether your tool actually recognizes what it's supposed to recognize. So when I was using this gold anti-teletonin antibody on the Western blot, it says maybe it sees red teletonin, but it also sees a whole lot of other stuff. So this is obviously not a good tool. Sometimes you can also come up with misleading conclusions because you're saving money or somebody else wants to save your money. So what I have for the last 20 years, I have always used the third from the top antibody. When I ordered a secondary anti-mouse, that was Cy3 conjugated. So this is the code number, this is the price, and it has made everybody very happy. Well, then, as it happens, I moved away from the coal face, and then somebody else had to take over the ordering. And actually, this person showed initiative and didn't just copy a code number, but checked out on the website and realized, oh, come on, there's a cheaper version around. Why does Elisabeth always spend so much money on our secondary antibodies? And then for the coming weeks, what we observed was that we had this sudden co-localization of all these mouse and rabbit antibodies, which, hadn't really been seen before. And the reason for that was that the cheaper one, you save some money, but it doesn't have the perk that this more expensive one has, which is minimum cross-reaction with rabbit serum proteins. And the serum protein is obviously an antibody. So this is the clean stuff that will not cross-react with your rabbit primary antibodies, whereas this stuff is suboptimal. If you want to combine anti-mouse and anti-rat, then you have to buy even more expensive secondary antibodies. So just by getting the wrong reagents, you can come to misleading conclusions. So what I would recommend that you do, always check your antibodies in Western blot, also commercial ones. These days, when you want to publish stuff, they will often ask that you validate commercial antibodies by knockdown experiments. Don't trust anybody. So collaborators can become victims of human error. So it has happened to me that people sent me stuff, and it wasn't necessarily meant that they just mixed up the labeling of the tubes. And luckily, this was the kind of reagent where it was pretty easy to spot which should be in which tube. And sometimes your collaborators are not as friendly as you think they are. So double check everything, every reagent that you're working with. And the third essential is that you always have to perform the appropriate control. So whenever you do your stainings, you have to have a negative control without primary antibodies, because the secondary antibodies, which is what you will be seeing in your fluorescence microscope, they may show all kinds of stuff. And I had, again, last week, I was contacted by somebody who said, oh, and in our cardiomyopathy mice, we have this relocation of our favorite protein to the plasma membrane, and can we show you the pictures? And then I looked at the pictures and I said, well, that's just the background from the secondary antibody, because you have cardiomyopathy mice, and this is probably, I would say, your primary is a mouse monoclonal. You have cardiomyopathy mice with fibrosis, so the anti-mouse secondary, it just shows this plasma membrane associated background. And that's most likely it was not less exciting, but just an artifact that result. So that's the reagents, but it's also, once you have produced your immunostain sample, you want to look at it. And in order to look at it, you have to know what actually do you want to look at? Do you want to have a closer look at it? Do you want to have an overview? And which is the best microscope in order to do that? Because unfortunately, nobody is perfect. No human is perfect, and also no microscope is perfect. And it doesn't matter how many millions it has cost. Each microscope is specialized for a particular tool, for a particular job. And if if you're using a bicycle on the motorway, it's no good. But for mountain biking, a Jaguar is probably not so much fun. So how do you decide it? So usually, there's it depends what you want from your sample. So is your sample dead? Is it fixed? Is your sample alive? And if your sample is alive, then for the majority of application, a bog standard fluorescent microscope will actually be the best choice, because cells are not really keen on living cells are not keen on being lasered by a confocal. So you have to go down with the laser power quite a bit in order to visualize living cells in a confocal. The other thing you have to bear in mind, the confocal microscope is rather slow in its scanning speed. So if you're looking, the whole point of looking at living cells is to look at dynamic stuff. And if you're looking at fast events in a conventional confocal, you're not going to be able to visualize that. So you will need a spinning disk confocal. The scale is important. Do you want to look at an entire heart? Do you want to get through to 200 micrometer imaging depth of your ventricular wall? Or do you want to have a high resolution with the 0.2 micron scale? So this will define whether you use a multiphoton or whether, again, you use a confocal. How many images do you need? Do you need a lot of images in order to make your conclusion from your experiment? Or are you happy if you have your 3 images that show what you want to show and the appropriate control? Again, this will define, so with an operator, you can take tons of images, and you can then do the quantifications and so on very easy. But if you look the images, at a higher resolution, they are not fabulous. So they give you a lot of information, but don't look at it too closely. Do you want to compare the colocalization of several things? So, do you want to have a look at more than one fluorochrome? Yes, then you will have to use, for colocalization, you will have to use a confocal microscope. Or are you happy just looking at one thing at a time? And Alexa 647 is a fluorochrome you like? Yes, then by all means use super resolution microscopy. And STORM, you also need the patience of a saint to use STORM. This is a different way of visualizing basically the same thing that I've taken from an article that I will show you the source for later. And again, it shows you that none of these beasts is perfect. So usually when you make your choice which toy to use, so it depends what kind of contrast, is sample alive, is it dead, which resolution, which scan speed do you need, and how deeply do you want to penetrate into your sample. And what you can see from these lines in blue is that a wide field is great for speed, it's great for living cells, but for resolution it's not so great. Whereas a confocal is obviously better for resolution, but the cells are not keen, and so on and so forth. You will have, in addition to antibodies, you will also have other tools in order to visualize your stuff. And so one stuff that is of interest to many people in biology to visualize is actin. And this is just a compilation of the ways how you can visualize your actin. And again, you have to bear in mind, what do I actually want to conclude from my experiment? What do I want to see? Because on the bottom image, you see MCF7 cells, which is some crap cell line that, it's not a cardiomyocyte. So as far as I'm concerned, it's an inferior cell line. But just when you compare what you visualize with the different actin tools, visualization tools, it becomes quite apparent that you're actually looking at different populations of the actin. So depending which tool you're using, you may preferentially stain, with the phalloidin, it's pretty clear you will only be staining the filamentous actin, but also with the other ones, it may preferentially stain some subpopulations. and it will, if you have a bulky tag sitting at the end, it will affect the dynamics as well. And the cell migration folks don't really like admitting it, but LifeAct does affect the dynamics of actin. The tools you will, in the 21st century, usually use in order to visualize your signal is fluorescence. So you will use a fluorochrome conjugated to your secondary antibody, and based on the rules of physics, they will be excited by a shorter wavelength, and they will emit at a longer wavelength. And so this is the poster boy or poster girl of fluorochromes. So this is Alexa 488, and it's often used as an example because it has this beautiful narrow excitation and this beautiful narrow emission peaks. Now this is not what fluorochromes always have. Because when you see here comparison of four of these Alexas, so in the different shades that work in combination, you can see, yes, the Alexa 488 has narrow peaks, but the other ones have bumps. And if these bumps overlap with the emission bump of some other fluorochrome you use, you may be having crosstalk between the fluorochromes, and again you may come to a misleading conclusion about co-localization and so on and so forth. So always check out your fluorochromes, check out when you're using something new. Occasionally, there may be a hillock far down the line that they don't necessarily tell you about in the catalog. Down here, you find a list of commonly used fluorochromes. So, again, as we said, we use a lower range excitation. We then detect a green signal, red signal in the eyepiece, and so the ones that I have circled here. So, if you have any choice, try to avoid those, because these belong on the history of fluorochromes. So that's the kind of stuff that while you're focusing, it fades, it bleaches. So if there are much better fluorochromes around these days, and it depends a bit on your personal taste. So I'm a fan of the Cy dyes, but so they are available from Jackson Immunochemicals. Actually, I'm a fan of Jackson Immunochemicals because their secondary antibodies are really, really good quality. Whereas, the Alexa 488 is also perfectly respectable fluorochrome. The antibody quality of Invitrogen is not as high often. And there's also the Dilight now these days. So these are very, very stable fluorochromes. So you don't have to worry about, doing your exposures in absolute darkness, because these dyes are very, very stable. But you have to consider the physical properties. And one of the physical properties where, it's the law of nature, is, so these are two different staining approaches. So we have always visualized tubulin, and on the left-hand side, visualized tubulin with Alexa Fluor 488, so in green. And on the right-hand side, we have used, the only difference is a different secondary antibody, which has a different fluorochrome. And you can see that, it's not quite as crisp. This is a particularly bad example in comparison, but that's just something that you may have to take into account, that for conventional fluorescence microscopy, the green is often your friend. The green is not your friend when you're looking at tissue that is necrotic, because that will have a lot of green autofluorescence. And then you may be better off using a red or dark red fluorochrome. You also have to know what can your microscope actually do. So for example, so here I've got the laser lines at the Thomas SP5 confocal microscope. And that's just what we can excite. And if you happen to use a secondary antibody that's conjugated to a fluorochrome that's in this void here. So, you know, Texas Red, Alexa 568, Alexa 594, you will not be able to excite it very, very well. And so therefore you will have wasted an experiment, because no matter that this beast cost 300 pounds when it was 1,000 pounds when it was bought, it will not be able to excite your fluorochrome. So when you plan your experiments, make sure that you know what the toy that you're actually then using for the imaging actually can do, because there are confocals around that have a line here in the middle as well. Which lens is best? That defined where you what kind of samples are you looking at? Are you looking at things in a plastic dish, you know, where you have a fat plate to go through, or are you looking at stuff where you have a cover slip and then you will be able to approach closely with your lens with an oil immersion microscope? There are mixed lenses around that have something that's called a correction color, which means you can use different immersion substances. So, for example, glycerol, water, or oil. But if the correction color is on the wrong position, it's not going to help you with the imaging either. And always be careful that for a high-end microscopy, you use a lens that has the maximum NA that you can get for your microscope. One thing which you will have to bear in mind more and more in the future. So, you know, up to now, a lot of people, they say, oh, and, you know, and then so we immunosensed and then we went to a microscope and that's it. But these days are probably over, because so this was published in the summer in eLife, and this is a complaint by this group, and it has got quite a bit of traction in the imaging field, and it asks efforts from researchers, funding agencies, and journals that you supply the information. So in a few years, I doubt very much it will be sufficient that you say, oh, well, and then we did fluorescence microscopy, but you will have to specify which lens did you use, which immersion fluid, and all kinds of parameters. So make sure that you already note that down now, so that you don't get caught out whenever you want to publish in eLife, for example, in 2 years. So in the olden days, when you wanted to see more, you had to use an electron microscope. And this is a beautiful picture from Pauline Bennett showing heart muscle tissue. And you see the nuclei here, you see the myofibrils, you see the mitochondria. The reason why the mitochondria look a bit have suffered a bit is that this is actually an electron micrograph of a human heart sample that had been sitting in liquid nitrogen for a while. So, I mean, it's amazing that it looks that good for what it experienced. But usually in order to get to an electron microscope, you have to do very, very drastic things to your samples. So you usually have to fix them with glutaldehyde, you have to dehydrate it, you have to embed it in resins and then cut it and so on. So, you know, this is brutal. And therefore, you know, immunoreactivity goes down the drain and so on and so forth. And therefore, for a long time, people had a desire to see more without doing these cruel things to their samples. And that's where super resolution microscopy comes in. So now this is Europe, yeah. It's this weird place across the channel. You can even see my hometown here, so that's lighting up. So that's Europe at night, but that's what you see when you don't do super resolution microscopy. But when you want to do super resolution microscopy, what you want to see the individual street lamps. And the problem that we have here is, so we have now our Alexa 488 fluorochrome, and I told you, that's about the best fluorochrome you can have from its properties. But when you're actually detecting the signal of one single fluorochrome on your CCD camera or your photomultiplier, whatever you use, you won't see that tiny dot, but you will be seeing something much bigger. And the difference between the two is the point spread function. Now, in the XY axis, we are not too unhappy because, you know, at least it goes in the same direction. But if we are looking vertically, so in the XZ direction, the point spread function looks even more sad. And that's actually an idealized version of it, because when we look at it in a proper way, you can see, so, you know, there's all these weird tails and of this signal, and it depends, you know, very much on the setup of your scope, how bad it will actually be. Now, the two main ways of I will only be discussing two ways of super resolution microscopy, and that is STORM and that is STED. So let's start with STORM. So STORM stands for Stochastic Optical Reconstruction Microscopy. So what you use is the fluorochromes have the ability to blink. So they will not always be emitting light, but they will be on and off and on and off. And it depends on the fluorochrome, it depends on the mounting medium, it depends all kinds of conditions. But what the uses of it is, so when we have here a dot, we have here two dots, and, you know, because of this point spread function, they're overlapping. And what you have with the STORM, because not both fluorochromes will be ideally active at the same time, if you know, take a very, very long exposure of all these on, off, on, off, on, off conditions, you will be able to resolve these two dots. Now, I already told you before, you need the patience of a saint for STORM. Because you have to take a long exposure in order to collect enough blinkers so you can do something. And this is again an image set given to me by Pauline Bennett of some myofibrils stained in and visualized in STORM. And this is my mesin M-band antibody. And there's two things that you see here. One is that there's a lot of starry sky as well, so, you know, so it's obviously some other blinking stuff going on. And the other thing that you see here is that this is a single labeled thing, so it's not nothing with quadruple or anything like that, because actually the only fluorochrome that will really be very, very reliable in STORM is Alexa 647. And so therefore, this is a definite disadvantage. And whenever people talk about multicolor STORM, what they're actually doing is they're stripping their samples, you know, like in the olden days with Western blots, boiling them in beta-mercaptoethanol or something cruel, and then re-exposing them to another set of primary and Alexa 647 secondary antibody. The second way of getting more information out of your sample by a super resolution microscopy is STED, so that's stimulated emission depletion microscopy. The creator of this was Stefan Hell. And what the idea he came up with was that basically in order to just visualize the spot, you use this donut in order to bleach the stuff around it. And so this is again visualized on this slide. So you have the donut, and because of the donut, you can separate the two samples. And again, I'm showing you that on a myofibril. So this is an epitope on titin T12, which is 120 nanometers apart, and, you know, in confocal, well, on a good day you can sort of guess where it is. So this is a particular bad confocal image, but it was just, you know, to show how superior STED is, where you can clearly show that this is a doublet actually. And again, also with STED, you have to be careful because not all fluorochromes, you can't just go with your box standard immunostandard sample and go to the STED and hope you will get better resolution. You also need dedicated secondary antibodies with dedicated fluorochromes to do STED. And there's only you know, in our hands, 2-color is possible, but yeah, the best one is the 647 range as well. But it is possible to correlate it with others. So we have perfect antibodies. We know which microscope to use, but now we have to prepare our samples, and we have to use our microscopes. And you know, there's more pitfalls. Unfortunately, you know, there are pitfalls at every corner with this technique. I mean, it's not unique in doing experiments. And so in what you usually want to do, because these immunostaining procedures are quite lengthy, and you know, you don't want to avoid diffusion of your antibodies and degradation of your proteins, you usually will carry out a step which is called fixation, and which basically takes a snapshot of your image. And you have two choices in how you want to fix your. You can cross-link your sample. For this, you will be using formaldehydes. So paraformaldehyde, which is a sort of general lab working horse. Don't use formalin because it contains methanol. Or if you want to use electron microscopy, or if you want to use look at microtubules, you will have to use a stronger fixative like glutaraldehyde. And a cross-linking means that you have excellent structural preservation, but you may have too much cross-linking, and you may have too much cross-linking, and the epitopes, your antibodies don't recognize the epitopes anymore, especially with glutaraldehyde. You also have to quench the sticky sites, so you have to be quite careful with those. And for the microtubules, you could also use paraformaldehyde in microtubule protecting buffer, by the way. So for initially, when you're, for example, validating newly produced monoclonal antibodies, you will usually not do cross-linking fixation, but you will use precipitation. So you will just do, for your screens, you will just do methanol fixation because it's more gentle. So that means more antibodies survive the screening and will work. And it's also, for crowded areas, it can be an advantage to use precipitation, but you know, you would never use a methanol or acetone fixed sample and go to it with it to the electron microscope, because it would just look like a graveyard. And all these solvent-based precipitation methods have to be used at -20. And, you know, depending whether your stuff sits on plastic or your stuff sits on glass, you can either use methanol or acetone. But if your antibody initially doesn't work at PFA, it's always worthwhile trying whether, you know, maybe with methanol you can sort of elicit a signal. But while I'm always keen on booze, booze is not always good. And for example, if you look at GFP transfected stuff, so here we have examples of GFP transfected cardiomyocytes, PFA fix, GFP channel, beautiful fluorescence, transfected cell, methanol fix. The GFP fluorescence is almost gone. But the GFP is still there, because if you use a primary antibody that recognizes the GFP, it's fine. So, you know, we can visualize it, but GFP and methanol is not a great combination. And just to remind people that, you know, if you have stringy filamentous stuff with the GFP, that can be a fixation artifact. So that may not necessarily be true. Also phalloidin. So, you know, phalloidin is the fluorescence reagent for idiots, but not for methanol fixing idiots, because it will always work with paraformaldehyde, but if you methanol fix your samples, you lose the phalloidin reactivity to a different extent as well, and then you just see these residual signals and no longer the entire filamentous actin as you would have seen it otherwise. Okay, so we have fixed our sample. What do we have to do? Yes. Well, next thing, what we usually, the majority of people that I talk to want to look at cytoplasmic stuff. So they are not interested in surface receptors or something, but they want to look at stuff inside of the cells. And then you have to make holes into your plasma membrane, because otherwise you don't get your primary antibodies in. And the two reagents that you have available to make your holes in your plasma membrane are Triton X100 and saponin. Now, Triton X100 is what the majority of you will have used. Again, it's one of these lab working horses, but it is a tricky customer. So you have to be careful with your Triton. Saponin is more gentle, but it's also sometimes a bit too gentle because it's also reversible. And on the next slide, you see an example that, just which detergent you're using can drastically affect this conclusion that you draw from your sample. So I've used, so this is just boring cost cells, but for this purpose they were good enough. And I've used antibody in red that recognizes the Golgi apparatus, and an antibody in green that recognizes the endoplasmic reticulum. And when I do Triton permeabilisation with the Golgi signal, it's okay. So it's on one side of the nucleus, it's blah, I'm not too worried about this one. But what I'm a bit concerned about is this ER signal. And what I'm massively concerned about is that I have all this signal in the nucleus, which I have counterstained with DAPI. And if I use exactly the same cells, exactly the same fixation, and the only difference is which detergent I use, suddenly with the Golgi, we see these textbook-like stacks of Golgi apparatus. And, this is relatively comparable. But with the endoplasmic reticulum, the picture has completely changed, because here we see something which, again looks like textbook endoplasmic reticulum, and all this nuclear shit has gone. And the only difference was the detergent. So be careful if you're looking at membranous organelles. Saponin is your friend. Also, if you're looking at autophagy, autophagosomes, and so on and so forth, Triton is not your friend. Then you have to put your reagents on your stuff, and then you have, depending what you're looking at, you have different incubation times. Now, this picture I wouldn't have been able to show to you without COVID. Because this experiment was forced upon me by the impending coronavirus-induced shutdown. So what I was doing at the time, I was, what I usually do, or what I often do, I was staining whole mount preparations of embryonic mouse hearts. So this is, 0.2 millimeters of heart. And you bath it in different antibodies. And I usually bath it in primary antibody at 4 degrees overnight, and usually bath it in secondary antibody at 4 degrees overnight. But because they were threatening to lock us up, I thought, 5 hours at room temperature may also be okay. Let's try it. We are desperate. Well, anyway, so what you see here is that for the phalloidin, which is a really, really tiny compound, the diffusion in 5 hours was no problem. So you see this homogeneous signal throughout this field, and the intensity for the phalloidin is completely the same on the outside of the heart and on the inside of the heart. Now, unfortunately, this was not the case for the secondary antibody. So for the Cy3 anti-mouse, you see this gradient of signal. And you know that the primary had no problem sitting there. So it got through because it had the chance to react overnight. But we are not seeing the primary antibody. We are only seeing the Cy3 that sits on the anti-mouse secondary antibody. And if we don't give it enough time to diffuse through, to react with all the primary antibody that's around, then we will have this gradient. And, again, it may lead to a misleading signal conclusion. And for example, also, if some of you may be staining adult red cardiomyocytes, and these fat bastards, they need a lot of incubation time, because otherwise you will just be staining hollow cores, because just that your antibodies don't get through in the middle of the cell. Now, when you're working, you have to work fast, and you have to work in a clean fashion, because dirt is also fluorescent. And I never thought I would be able to show this thing, but then I had the slowest project student ever, And he managed to work so slowly that we had drying out artifacts in our samples. And so you get these interesting structures, which, could be biological, but actually it's just a drying out artifact. And it's a drying out artifact that can be visualized in all four channels that we can excite in this particular microscope. So make sure that your sample doesn't dry out during the staining. Other reasons for background is something that is inherent in the biology, and there's not much you can do against it. And when I look at a human heart sample, I can tell you whether this heart sample is from a patient that is younger than 40 or older than 40. Because if you have a 23-year-old heart, then you see in this particular case, we have stained myomesin in green stripes, we have stained for laminae in red, and we have stained for the nuclear on this side with DAPI. But once you reach the watershed of 40, because we are dealing with a post-mitotic tissue, the cells accumulate a lot of rubbish. And, the scientific name for rubbish in a cardiomyocyte is lipofuscin granula. And they just have them. And, that's just something that you will also see in the negative control. And that's just something that we have to live with. And it's absolutely fine, as long as you're not proposing that there's this funny interaction going on between laminae and myomesin in the older patients, because you have the co-localization and the yellow signal, it's fine. So you just will have to live with this. So we have done all our antibody incubations, and now we are almost there at the microscope, and we are using our mounting medium and our cover slips, and almost ready to go. And there's another pitfall, because not all mounting media are good mounting media. So these guys obviously were not very successful with one. So Vector Shield is a commercial mounting medium that you can buy in a pot. Prolonged diamond is also something that you can buy in a pot. Now, prolonged diamond looks pretty all right. Vector Shield is obviously not something where I would either buy the pot or I would buy shares in the company. Now, I don't know what happened to old glycerol. So old glycerol was one of the bottles that I had standing around. Old glycerol has a dramatic problem because old glycerol has some nuclear background in all channels. So that's very, very peculiar. Whereas new glycerol is almost as good as prolonged diamond. And I'm, I don't like spending money on commercial products if I don't have to. So, I for mounting is I use new glycerol with M-propyl gallate to mount my sample, so it's homemade stuff. And this is not called after me, Elisabeth. So that was a Swiss researcher who tested all kinds of different combinations many years ago now, 35 years ago, something like that. And then they came up with this recipe that I have been taking home from Switzerland. And as I said, it's glycerol tris buffered with M-propyl gallate. And I was happily using this mounting medium for years and years and years and years until M-cherry came along. And then I had to realize that Elisabeth doesn't work with M-cherry, because while you're focusing your M-cherry that you have still looked at happily in your living cell, it disappears, the signal disappears. So you have to use a different mounting medium. So in my case, I tend to use cured polyvinyl alcohol with M-propyl gallate in order to visualize cherry, whereas, GFP is absolutely no problem with Elisabeth's mounting medium. In order to prevent fading of fluorochromes, what people often add is these anti-fading reagents. So the one that I tend to add is N-propyl gallate. There are others around. So phenylene diamine is one, for example, that is extremely efficient. But unfortunately, it's not compatible with cybidize. So that's not, you can't use those two in combination. TDE is a mounting medium that's often used by super resolution microscopists, which is unfortunately incompatible with Alexa dyes. I'm not a believer in DAPCO, so it doesn't matter whether you put it in or you leave it out. So in my experience, it's pretty useless, but well, maybe it's just me. And you also have to, so usually you mount your sample with a cover slip. And if you have any intention of doing super resolution microscopy, you have to make sure you use the appropriate thickness of cover slips. So you can't just take any box from the drawer and, slap it on. But you have to make sure that you're using 170 nanometer tested 1.5 thickness cover slips. Bleed-through. Well, bleed-through is another enemy that we have, and that's something that comes from the microscopes. And the problem with bleed-through is that when in the olden days you used a conventional, or you still use a conventional fluorescent microscope, and usually the bleed-through goes from the red channel into the green channel, because there's these idiots around who buy the cheap green filters, so they are blue and not turquoise, and they don't cut off sufficiently against the red fluorochromes, which is why a lot of people historically used to stain the red and overstain the green. And then they take the sample to the confocal microscope, and unfortunately it's the other way around, because you have often bleed through from the green to the red channel. And this is demonstrated on this. So this is an old software, but just to make the point, it doesn't make a difference. We have visualized myosin binding protein C here with our antibody in 488 nanometer excitation, beautiful doublets. We are happy. In 543, when we are collecting signal, we are also seeing something, which we shouldn't, because when I go over here and look whether the laser line is actually active, it's 0. So there shouldn't be any signal coming through here. So the signal that I'm seeing here is the bleed through that comes from the green channel into the red channel. And this is a real problem with confocal microscopes if you have a very, very intense green signal. And this is why that the majority of confocal microscopists will recommend that you scan your sample sequentially, so that you don't scan the green and the red at the same time, but that. And again, you can come to completely erroneous conclusions, because if you scan them at the same time, you would assume that, okay, so we have yellow, we have colocalization, so myomesin is a protein of the intercalated disc. Now, since 1984, when myomesin was first identified, it was never seen at the intercalated disc. So, this is just something that the confocal microscope has invented because you haven't used the appropriate imaging conditions. Whereas if you use sequential mode, they're beautifully separated and there's no problem at all. Now, but often, you're not just after your pretty pictures. You may wish to get numbers out of them. And if you want to get numbers, so quantify your fluorescence, that's, you're thinking you have problems so far. Well, think again. Because quantifying fluorescence is a completely different bag of stuff and has its own sort of problems. And one of the problems is photobleaching. So, it depends how long you take to focus your sample. If you're slow or you don't find stuff, after 60 seconds, especially if you don't use anti-fading reagent or the wrong mounting medium, you may not find much of your fluorescence left. And you can imagine, if you quantify this compared to quantify this, you could come up with all kinds of rubbish. Sometimes the quantification is marred by very, very unexpected problems. Now, this is a confocal microscope that lived in the Rockefeller imaging facility. And this was a pampered confocal microscope, checked all the time, and only run by expert staff. But what they eventually realized was that with the 488 nanometer laser, they had a bit of a problem with laser fluctuations, and over time. So and it didn't necessarily matter how long the laser was running. And you can again imagine that, if you measure your stuff here or you measure your stuff here, it may affect your numbers. Illumination non-uniformity. So, you know, if you do conventional fluorescence microscopy and the lamp is not properly adjusted, again, you know, if you get your number from the periphery or you get your number from the center, it will affect your number. So you have to be careful about that. And that's all kinds of pitfalls that can happen when you want to quantify fluorescence. But, you know, let's be optimistic and let's assume you have done all the controls. You have made sure that your quantification is as good as possible and that your raw data is close to perfection. And follow the instructions that is in this tutorial. So Alison North, you may see some overlap in the opinions and prejudices in this piece, because Alison North, who is the head of the Rockefeller imaging facility, got her training in microscopy in the same lab where I got it. So there's a definite overlap in attitudes. So I would recommend the reading of this article to anybody who has any plans to do these kind of experiments. But once you have your perfectly imaged samples, only then Dr. Holt will see you. And so Mark Holt is the new school lead for image analysis. So this is a title he sort of came up with himself because it stands for liar. I don't know why, but he is an expert in getting numbers out of fluorescence data, and I assume he will give a presentation on his expertise at some point later in this series.